Imagine you could hold your entire genetic code in your hands. All 3.2 billion letters of DNA that make you uniquely you, the instructions that built your body from a single cell, that determine your eye color, your height, perhaps even aspects of your personality. If you printed it out, letter by letter, it would fill approximately 200 telephone books. Yet this vast encyclopedia fits inside nearly every cell in your body, in a space so small it's invisible to the naked eye. But here's what scientists have recently discovered that changes everything. We've been reading only about 1.5% of those books. The rest? For decades, we called it junk DNA. Genetic gibberish, evolutionary leftovers, meaningless code. We were spectacularly wrong. That so-called junk? It might contain the most important information of all. Hidden powers that could revolutionize medicine, explain consciousness, unlock extraordinary human abilities, and even redefine what it means to be human. Your DNA holds secrets we are just beginning to understand, and what we're finding challenges everything we thought we knew about human potential. In 2017, Dr. Diana Evans was analyzing the genome of a patient with an inexplicable immunity to several viral infections. What she found hiding in the supposedly non-coding regions of his DNA would upend decades of genetic understanding and open the door to capabilities we once considered impossible. I remember staring at the sequence and thinking this cannot be right, Dr. Evans recalled. This section wasn't supposed to do anything, yet here it was, clearly activating during viral invasion, producing a protein we'd never seen before. That discovery was just the beginning. From ancient viral DNA that protects us against disease to dormant genes that could extend human lifespan by decades. From hidden neural connections to unprecedented regenerative abilities. Your genome harbors extraordinary potentials that are only now coming to light. And as we unlock these secrets, we face profound questions about our future. What happens when we can access these abilities? What will it mean to be human when we can rewrite our own genetic code? The story of your DNA and its untapped potential begins not with the famous double helix discovery of 1953, but much earlier with a quiet monk tending his pea plants, making observations that would take over a century to fully appreciate. In the garden of an Augustinian monastery in what is now the Czech Republic, Gregor Mendel meticulously crossed and documented thousands of pea plants between 1856 and 1863. His work established the basic rules of heredity, showing how traits pass from generation to generation in predictable patterns. But Mendel had no concept of DNA. He couldn't see the actual mechanism of inheritance, only its effects. When scientists finally identified DNA as the molecule of inheritance in the early 20th century, they faced an enormous challenge: deciphering its language. The discovery of the double helix structure by Watson and Crick in 1953 was revolutionary, but it was just the beginning of understanding what DNA actually does. The structure immediately suggested how DNA might replicate itself, but not how it created us, explains Dr. James Langley, geneticist at the Cambridge Institute for Medical Research. That required breaking the genetic code, understanding how those four simple chemical bases, arranged in endless combinations, could possibly build something as complex as a human being. By the 1960s, scientists had cracked the basic code of how DNA creates proteins, the workhorses of our cells. They discovered that genes, sections of DNA, provide instructions for building proteins, which then carry out virtually all cellular functions. This central dogma of molecular biology, DNA to RNA to protein, seemed to explain how genetics works. But there was a problem, a massive problem. When the Human Genome Project completed its first draft in 2001 after 13 years of international effort, it revealed something unexpected. Humans have approximately 20,000 protein-coding genes, far fewer than expected. Even more surprisingly, these genes make up less than 2% of our total DNA. It was like discovering that only 2% of a massive instruction manual actually contained instructions, says Dr. Langley. What was the other 98% doing? Was it just filler? Evolutionary garbage that accumulated over millions of years? That's what many scientists initially thought. They called it junk DNA, a term coined in the 1970s that persisted for decades. But even as this dismissive label took hold, some scientists remained skeptical. Why would evolution, notorious for its efficiency, preserve so much useless material? Why would our cells expend precious energy replicating billions of junk letters every time they divide? The answer would begin to emerge in 2003 when the ENCODE project, Encyclopedia of DNA Elements, launched with a bold mission: to identify all functional elements in the human genome. Their preliminary findings in 2007 hinted at something revolutionary, but it was their 2012 publication that shook the foundations of genetics. ENCODE researchers announced that at least 80% of the human genome was biochemically active, not junk at all. Much of this activity occurred in the vast non-coding regions previously dismissed as meaningless. This revelation raised a profound question that would launch us into the next frontier of genetic discovery. If most of our DNA isn't building proteins directly, what exactly is it doing? The answer would prove far more fascinating and potent than anyone imagined. 20 miles north of San Francisco, in a laboratory overlooking the Pacific Ocean, Dr. Diana Evans peers into a microscope at what appears to be an ordinary blood sample. But the cells she's examining are anything but ordinary. These cells contain a genetic sequence that shouldn't exist in humans, she explains, her voice barely containing her excitement. It's a sequence we found in less than 0.01% of the population, hidden within what we once called junk DNA. And it appears to provide complete immunity to at least 7 different viruses, including some forms of influenza and herpes. What Dr. Evans discovered in 2017 was a functional gene sequence buried within non-coding DNA, a sequence that produces a unique antiviral protein when certain infections are detected. More remarkably, this sequence appears to have entered the human genome through an ancient viral infection approximately 300,000 years ago. Viruses sometimes insert their genetic material into our DNA. It's called a retroviral insertion, Dr. Evans explains. Most are harmless relics, but this one appears to have been weaponized by our genome to fight the very types of organisms it came from. It's like evolutionary judo, turning a virus's strength against itself. This discovery represents a profound shift in how we understand our genome. Much like dark matter in physics, which makes up the majority of our universe but cannot be directly observed, the non-coding regions of our DNA exert enormous influence despite being invisible to our earlier methods of analysis. Consider this. While your protein-coding genes have remained relatively stable throughout human evolution, these dark matter regions have evolved much more rapidly. They contain the majority of genetic differences that separate you from a chimpanzee, despite our 98.8% genetic similarity in coding regions. More astonishingly, when scientists at the University of California performed a detailed analysis in 2019, they discovered that many non-coding regions are under stronger evolutionary pressure than coding genes, meaning nature is actively selecting for changes in these supposedly junk areas. This suggests something rather extraordinary, says Dr. James Langley. The most important parts of our genome might not be the genes we've been studying for decades, but rather the regulatory elements that control them. These regulatory elements, stretches of DNA that determine when, where, and how much proteins are produced, act like the conductors of a vast genetic orchestra. They decide which instruments play, when they come in, how loudly they perform, and when they fall silent. And they vastly outnumber the instruments themselves. In 2018, the discovery of enhancer elements, non-coding regions that can amplify gene expression from considerable distances, revealed another layer of complexity. These enhancers don't just turn genes on or off. They can dial their activity up or down with remarkable precision, responding to environmental cues, developmental stages, or cellular needs. It's as if we've been focusing on the individual musicians, not realizing there was a conductor at all, explains Dr. Langley. And now we're discovering there isn't just one conductor, but hundreds of thousands of them, all coordinating different sections of the orchestra at different times. This explains a puzzling observation. How can humans be so complex with only 20,000 genes, when a seemingly simpler organism like rice has 40,000? The answer lies not in the number of genes, but in the sophistication of their regulation, the complex interactions of enhancers, silencers, promoters, and other regulatory elements that precisely control gene expression. But even this doesn't capture the full extent of your DNA's hidden powers. Because beyond regulation lies something even more extraordinary. Vast networks of RNA molecules transcribed from non-coding DNA that don't create proteins but perform crucial cellular functions on their own. We've discovered over 80,000 RNA sequences produced from non-coding regions, says Dr. Evans. Many act as cellular switches, turning processes on or off. Others regulate gene expression at various levels. Some even appear to retain memories of past environmental exposures, potentially explaining how experiences might be passed between generations. This last point refers to the emerging field of epigenetics, changes in gene activity that don't involve alterations to the genetic code but can still be passed down to at least some future generations. These changes often occur in non-coding regions, suggesting they may be the primary interface between our environment and our genome. In 2020, researchers at Emory University demonstrated that mice exposed to specific trauma developed distinct epigenetic markers in their sperm, changes that appeared to transmit fear responses to specific stimuli to their offspring, despite those offspring never experiencing the trauma themselves. We're beginning to understand that your genome isn't just a static blueprint, Dr. Evans explains. It's a dynamic, responsive system that can adapt to environmental challenges and potentially pass those adaptations to future generations. But if our non-coding DNA holds such power, if it contains antiviral protections, regulatory networks, RNA machinery, and epigenetic memory systems, what other capabilities might be hidden in these previously overlooked regions? The answer brings us to perhaps the most tantalizing frontier in genetic research, our dormant evolutionary inheritance. On a crisp September morning in 2022, in a high-security laboratory at Harvard Medical School, something unprecedented occurred. A team led by Dr. Jacob Lawson activated a gene sequence that had lain dormant in the human genome for approximately 65 million years. The gene, dubbed Phoenix 1, appears to be a remnant from our evolutionary past, a functional sequence inherited from distant ancestors but silenced over millions of years of evolution. Its function, extraordinary tissue regeneration capabilities far beyond what modern humans possess. We identified Phoenix 1 by comparing human genomes with those of animals known for remarkable regenerative abilities. Dr. Lawson explains. Salamanders can regrow entire limbs. Certain fish can repair heart damage with no scarring. We hypothesized that mammals, including humans, might retain some of these abilities in our DNA, just in inactive form. After identifying several candidate sequences, Dr. Lawson's team focused on one that showed signs of having been active in mammals approximately 65 to 70 million years ago, roughly when dinosaurs went extinct and mammals began their evolutionary expansion. Using CRISPR-Cas9 gene editing technology, we were able to reactivate this sequence in lab-cultured human cells. Dr. Lawson continues. The results exceeded our expectations. The cells demonstrated regenerative capabilities approximately 17 times greater than normal human cells, including the ability to rebuild complex cellular structures that are typically permanent in humans. This discovery suggests something profound. Many abilities we associate with other species may actually lie dormant within our own DNA, evolutionary ghosts waiting to be reawakened. The implications are staggering. Beyond regeneration, researchers have identified silenced genes potentially connected to enhanced night vision, oxygen efficiency, hibernation capabilities, and even longevity pathways active in species that live far longer than humans relative to their size. We found what appears to be a functional hibernation sequence, explains Dr. Diana Evans, who collaborated with Dr. Lawson on a follow-up study. It's almost identical to sequences used by bears and certain squirrel species to lower their metabolic rate and survive without food or water for months. In humans, this sequence is present but silenced by specific methylation patterns that prevent its activation. Methylation, the addition of methyl groups to DNA molecules, is one of several epigenetic mechanisms that can silence genes without altering their sequence. It's part of the complex regulatory system that determines which genes are active in which cells at which times. But why would evolution silence potentially beneficial abilities like tissue regeneration or hibernation? The answer reveals a fundamental principle of evolutionary biology. Every ability comes with costs. Enhanced regeneration requires enormous energy and creates cancer risks, explains Dr. Lawson. Hibernation capabilities would have reduced alertness to predators. Enhanced sensory abilities often come at the expense of others. Evolution isn't about maximizing every trait. It's about optimizing the overall package for specific environmental challenges. This suggests that many dormant abilities in our genome weren't eliminated because they were useless, but because the cost-benefit calculation changed as our species evolved. However, in a modern context where we can potentially manage those costs through medicine and technology, reactivating these dormant capabilities becomes possible. Perhaps even more fascinating is the discovery that many of these dormant genes appear to retain their functionality despite millions of years of disuse, as if they've been deliberately preserved. We expected significant degradation in these sequences. Dr. Evans notes, when genes aren't used, they typically accumulate mutations since there's no selective pressure maintaining them. But many of these dormant genes show signs of negative selection, evolutionary pressure against mutation, suggesting they still serve some purpose we haven't yet identified. One theory is that these genes remain functional because they're occasionally reactivated during extreme stress, acting as evolutionary emergency equipment that provides survival options during catastrophic events. Some evidence supports this hypothesis. During cases of extreme hypothermia, some humans spontaneously enter a hibernation-like state with drastically reduced metabolism that allows survival in conditions that would normally be fatal. Similarly, there are documented cases of heightened strength, sensory perception, or endurance during life-threatening emergencies. These dormant adaptations may serve as a kind of evolutionary insurance policy, suggests Dr. Lawson. Capabilities that can be rapidly reactivated if environmental conditions suddenly change. This concept of rapid adaptation has gained support from studies of epigenetic inheritance. The idea that environmental experiences can create lasting changes in gene expression that persist across generations without altering the DNA sequence itself. In 2014, researchers at New York University found that mice exposed to specific odors alongside electric shocks developed a fear response to those odors. Remarkably, their offspring, despite never experiencing the shocks themselves, showed the same fear response and corresponding changes in their olfactory neural pathways. What's fascinating is that these changes occurred in regions previously considered junk DNA, says Dr. Evans. The non-coding regions appear to be where much of this environmental responsiveness is encoded. This suggests another hidden power of your genome. It may contain a sophisticated environmental memory system, one that can capture and preserve responses to environmental challenges and potentially transmit them to future generations through epigenetic mechanisms. But perhaps the most extraordinary dormant capability in our genome relates not to physical abilities but to our neurological potential pathways connected to consciousness, perception, and cognition that we're only beginning to understand. In 2023, neurogeneticists at Stanford University identified a network of regulatory elements in non-coding DNA that appears to influence neural plasticity, the brain's ability to form new connections and adapt to experiences. When experimentally enhanced in animal models, these elements produced substantially increased learning capacity and cognitive flexibility. These sequences appear to regulate how easily neurons form new connections, explains Dr. Langley, who was not involved in the Stanford study but has reviewed the findings. They essentially control the brain's balance between stability and plasticity, between maintaining existing knowledge and adapting to new information. More intriguingly, variations in these sequences correlate with certain cognitive abilities in humans, suggesting that much of what we consider intelligence or talent may be influenced by regulatory elements in our non-coding DNA rather than by the genes that build our neural architecture. This brings us to perhaps the most profound frontier in genetic research, understanding how our DNA shapes our minds, our consciousness, and potentially even phenomena we've traditionally considered beyond scientific explanation. At the Massachusetts Institute of Technology's McGovern Institute for Brain Research, neuroscientist Dr. Diana Evans leads a team investigating one of science's greatest mysteries: how consciousness emerges from the physical brain and how our DNA shapes this process. For decades, we've known that consciousness must have some biological basis, Dr. Evans explains, but identifying the specific mechanisms has been extraordinarily challenging. Recent discoveries in non-coding DNA may finally provide some answers. In 2021, Dr. Evans and her team identified a network of regulatory elements they've termed the Consciousness Regulation Network, CRN, a vast system of non-coding DNA sequences that appears to coordinate activity across various brain regions associated with conscious experience. The CRN doesn't build the brain's physical structures, Dr. Evans clarifies. Rather, it orchestrates how those structures communicate with each other, particularly in ways associated with self-awareness, attention, and subjective experience. What makes this discovery particularly remarkable is that the CRN shows unusually high conservation across species known to demonstrate self-awareness, from humans and great apes to elephants, dolphins, and certain birds, despite these lineages having diverged millions of years ago. This suggests that consciousness, at least in its basic form, emerged very early in evolutionary history and has been carefully preserved, notes Dr. Evans. But what's truly fascinating is how the network has expanded and specialized in humans. Human CRN sequences contain approximately 72,000 regulatory elements, nearly 3 times the number found in chimpanzees, our closest evolutionary relatives. Many of these human-specific elements appear to regulate genes involved in processing abstract concepts, symbolic thinking, and self-reflection. It appears that much of what makes human consciousness distinct isn't in our neurons themselves, but in how those neurons communicate, patterns orchestrated by regulatory elements we previously dismissed as junk DNA, Dr. Evans explains. This discovery has profound implications for understanding variations in human consciousness, including extraordinary mental abilities that have long puzzled scientists. Consider the case of Daniel Tammet, who can perform complex mathematical calculations instantly, learn new languages in days, and recite pi to 22,514 decimal places. Or Jill Price, who can recall virtually every day of her life since age 14 in minute detail. Or Stephen Wiltshire, who can draw entire cityscapes from memory after seeing them just once. We've long known that such abilities aren't explained by brain size or structure alone, says Dr. James Langley. Now we're finding that many savant abilities correlate with specific variations in non-coding regulatory regions that alter how existing neural networks function. In other words, extraordinary mental abilities might not require extraordinary brains, just unusual regulatory patterns that unlock capabilities most of us possess but cannot access. This concept extends to more common experiences as well. In 2022, researchers at the University of California, San Diego identified specific regulatory elements that activate during meditation and appear to facilitate the altered states of consciousness reported by experienced meditators. These elements show increased activity during deep meditative states, explains Dr. Langley. They appear to modulate the default mode network, brain regions active when the mind is at rest, allowing practitioners to experience consciousness differently than in ordinary waking states. More controversially, similar regulatory patterns have been observed during experiences traditionally considered mystical or spiritual, suggesting that capacities for transcendent experience may be built into our genome. We're not making claims about the ultimate nature of these experiences, Dr. Evans emphasizes, but we can say with increasing confidence that our DNA contains regulatory networks that can fundamentally alter how we experience consciousness, including states that feel transcendent or connected to something beyond ourselves. This research has also shed light on another mysterious aspect of human experience: dreams. In 2023, a team at the Allen Institute for Brain Science identified regulatory elements that activate specifically during REM sleep, the phase associated with vivid dreaming. These elements appear to facilitate neural activity patterns that simulate experiences in the absence of external stimuli, explains Dr. Langley. They essentially allow the brain to generate its own reality, complete with sensations, emotions, and narratives, entirely from internal resources. More remarkably, variations in these regulatory elements correlate with differences in dreaming experiences. From individuals who rarely remember dreams to those who report lucid dreaming, the ability to become aware within dreams and sometimes control their content. What this suggests is that dreaming itself, one of the most mysterious aspects of human experience, is facilitated by regulatory networks in non-coding DNA, says Dr. Langley. And the variations in these networks help explain why our dream experiences differ so dramatically from person to person. But perhaps the most profound implication of this research relates to the potential for expanding human consciousness beyond its current limitations. If our subjective experience is largely shaped by regulatory elements rather than physical brain structures, could we potentially access different states of consciousness by altering these regulatory patterns? This is where the research becomes both exciting and ethically complex, notes Dr. Evans. We're beginning to understand how to temporarily alter these regulatory networks through various methods, from electrical stimulation and neurofeedback to certain psychoactive compounds and meditation practices. Early research suggests that such alterations can indeed produce profound changes in conscious experience, including enhanced sensory perception, altered time perception, decreased self-referential thinking, and increased feelings of connection or unity with one's surroundings. These aren't just subjective reports, Dr. Evans emphasizes. We're seeing measurable changes in brain activity, information processing, creative problem-solving, and even certain forms of intelligence when these regulatory networks are temporarily reconfigured. This raises profound questions about human potential. If our default state of consciousness represents just one configuration of regulatory networks among many possibilities, what other forms of experience might be accessible? And what capabilities might those alternative states unlock? The answers to these questions bring us to the frontier where genetics meets technology, and where the future of human capability may be defined. In a sleek laboratory in Boston's Innovation District, Dr. James Langley adjusts settings on what appears to be an ordinary blood testing device. But this machine, roughly the size of a microwave oven, represents a revolution in personalized genomics. 5 years ago, a complete genome analysis cost about $1,000 and took several days. Dr. Langley explains, this device can sequence your entire genome in under 3 hours for approximately $200. But more importantly, it doesn't just read your genetic code. It interprets it, identifying variations in both coding and non-coding regions and what they might mean for your health, abilities, and potential. This technology represents the vanguard of what many experts call the genomic revolution, a transformation that will bring the hidden powers of DNA from research laboratories directly into our daily lives. Consider the implications for medicine alone. Traditional medicine typically treats patients based on population averages, prescribing the same medications and dosages to people with similar symptoms, despite their genetic differences. It's like having one size of shoes for everyone, Dr. Langley notes. Sure, they might technically fit most people, but they'll be perfect for very few. Pharmacogenomics, the study of how genes affect drug responses, has revealed that variations in both coding and non-coding DNA dramatically influence how individuals respond to medications. Some process drugs too quickly, rendering them ineffective. Others metabolize them too slowly, causing dangerous side effects at standard doses. Still others lack receptors that certain drugs target, making them completely non-responsive to treatments that work well for others. We've discovered that up to 95% of people have genetic variations that affect their response to at least one common medication, says Dr. Diana Evans. And many of these variations aren't in the protein-coding genes we've focused on historically, but in the regulatory elements that control their expression. By analyzing these variations, physicians can now prescribe medications and dosages tailored to each patient's genetic profile, potentially eliminating adverse drug reactions that currently hospitalize over 2 million Americans annually and cause approximately 100,000 deaths. But personalized genomics extends far beyond medication responses. It's revealing how variations in non-coding DNA influence disease susceptibility, potential lifespan, cognitive tendencies, athletic capabilities, nutritional needs, and even psychological traits. We're discovering that many chronic conditions develop through complex interactions between multiple genetic factors and environmental exposures, explains Dr. Evans. By identifying an individual's specific risk patterns, we can recommend personalized preventative measures decades before symptoms might appear. For example, researchers have identified regulatory networks that influence cellular aging processes, potentially allowing interventions that could extend healthy lifespan by 15 to 30 years based on individual genetic profiles. These aren't blanket recommendations like exercise more or eat healthier, Dr. Langley emphasizes. They're precise interventions based on your unique genetic landscape, specific nutrients, environmental factors, behaviors, and even medications that interact with your particular regulatory networks to optimize function and prevent disease. Beyond health applications, this technology is revealing genetic factors underlying human performance and abilities, from cognitive strengths to emotional resilience to physical capabilities. We're identifying regulatory networks that influence everything from mathematical reasoning to verbal processing to spatial awareness, says Dr. Evans. Not to label people or limit them, but to help them understand their natural strengths and potential growth areas. This understanding is already transforming education in some forward-thinking institutions. Rather than forcing all students through identical learning pathways, these schools are using genetic insights to tailor educational approaches to individual learning profiles. Some students process information visually, others verbally. Some learn through abstract concepts, others through concrete examples. Some need frequent breaks, others sustained focus periods, explains Dr. Langley. Much of this variation appears connected to regulatory networks in non-coding DNA that influence neural processing. By aligning educational methods with genetic learning profiles, these schools are seeing dramatic improvements in both academic performance and student well-being, raising questions about whether our traditional one-size-fits-all education system may actually be suppressing human potential rather than developing it. Similar applications are emerging in athletics, where genetic analysis can identify not just physical aptitudes but optimal training approaches, recovery needs, injury risks, and nutritional requirements specific to each athlete's genome. We're moving beyond simplistic ideas like, find what you're good at, to something much more sophisticated, says Dr. Evans. It's about understanding your genetic landscape and learning how to work with it optimally, whether in education, career, relationships, or personal development. But perhaps the most revolutionary aspect of this technology isn't in analyzing our existing genetic capabilities, but in potentially enhancing them. In 2019, He Jiankui shocked the world by announcing he had created the first genetically edited babies, twin girls with a modified gene intended to provide HIV resistance. His work was widely condemned for crossing ethical lines around human germline modification without adequate safety testing or consent processes. However, more carefully considered applications of genetic modification are advancing rapidly. In 2021, the FDA approved the first CRISPR-based treatment for sickle cell disease, editing patients' blood stem cells to activate a gene that produces fetal hemoglobin, effectively curing the condition. Unlike controversial germline modifications that affect future generations, these somatic cell therapies modify only the treated individual's non-reproductive cells, addressing ethical concerns about permanent alterations to the human gene pool. We're now developing targeted therapies that can activate beneficial dormant genes, optimize regulatory networks, or temporarily modify epigenetic patterns to address specific conditions, explains Dr. Langley. These approaches don't alter your fundamental genetic code. They simply help you access capabilities already present in your DNA. For example, researchers have identified methods to temporarily activate tissue regeneration pathways, similar to those used by salamanders, potentially allowing humans to heal injuries that would normally leave permanent damage. Others are exploring temporary activation of genes that increase resistance to radiation, enable more efficient oxygen utilization, or enhance specific cognitive functions. These aren't superhuman abilities, Dr. Evans emphasizes. They're capacities already encoded in our DNA, just not normally accessible. We're simply learning how to temporarily access them when beneficial. This concept of genetic potential unlocking, rather than fundamental genetic alteration, represents a middle path in the often polarized debate around genetic enhancement, one that acknowledges both the remarkable capabilities hidden within our existing genome and the ethical importance of proceeding cautiously with modifications that could have unintended consequences. It also raises profound questions about how we define normal human capabilities. If our DNA contains dormant abilities far beyond what we typically access, perhaps what we consider normal is actually a highly restricted subset of our true potential. We're discovering that human capability is far more variable and expansive than we previously understood, notes Dr. Langley. The differences between what we consider ordinary and extraordinary often aren't in the genes themselves, but in how those genes are regulated and expressed. This perspective shifts our understanding of human potential from a fixed, predetermined quality to something far more dynamic and developable. A spectrum of possibilities encoded in our DNA, waiting to be accessed through the right combinations of knowledge, technology, and ethical wisdom. As we stand at this frontier, we face profound questions about how to proceed. How do we ensure these technologies benefit humanity broadly rather than creating new forms of inequality? How do we balance innovation with safety? How do we preserve human diversity while addressing suffering? The answers will emerge not just from scientific laboratories, but from broader societal conversations about our values, our future, and what it means to be human in an age when the boundary between what is given and what is chosen becomes increasingly fluid. As dusk settles over Cambridge, Dr. Diana Evans walks through Harvard Yard, reflecting on the journey that brought her to this pivotal moment in human history. 20 years ago, when she first entered genetic research, the human genome was largely considered decoded, a solved puzzle with just a few mysterious pieces. "We were so wrong," she says quietly. "We hadn't solved the puzzle. We'd barely glimpsed its true complexity." Today, Dr. Evans leads an international consortium of researchers focused on what they call genome activation therapy, techniques designed to safely access beneficial capabilities encoded in non-coding DNA without permanent genetic modification. What we're discovering challenges our most fundamental assumptions about human limitations, she explains. Much of what we've considered fixed biological constraints may actually be regulatory choices, evolutionary compromises that prioritized certain capabilities over others based on ancient survival needs. The consortium's work has already produced remarkable results. In 2022, they developed a therapy that temporarily activates dormant tissue regeneration pathways in patients with severe injuries. Unlike conventional treatments that simply support the body's existing healing capabilities, this approach awakens regenerative processes normally inactive in adult humans. A patient with a spinal cord injury that would traditionally cause permanent paralysis regained nearly full function after treatment, Dr. Evans notes. Not because we added anything new to his genome, but because we temporarily activated capabilities already present in his DNA. Capabilities similar to those that allow certain animals to regenerate complex tissues. Similar approaches have shown promise for conditions ranging from neurodegenerative diseases to metabolic disorders to traumatic brain injuries. Not by altering the genome, but by changing how existing genetic material is expressed. We're essentially developing a language for communicating with our DNA, explains Dr. James Langley, who collaborates with Dr. Evans on several projects. Learning how to ask it to express different aspects of its vast capability repertoire depending on current needs. This concept of working with our existing genome rather than modifying it represents an important shift in perspective. From viewing DNA as something to be rewritten to seeing it as a vast resource to be understood and utilized more fully. The human genome contains approximately 3.2 billion base pairs, Dr. Langley notes. If each base pair were a letter, that's roughly equivalent to 1 million pages of text. For decades, we focused on just the equivalent of chapter titles and section headings, the protein coding genes that make up less than 2% of that text. We're only now beginning to read and understand the full narrative. This narrative includes not just instructions for building proteins, but an elaborate regulatory system that determines which capabilities are expressed when, where, and to what degree, a system that appears to have preserved extraordinary capabilities from our evolutionary past while keeping them dormant until needed. Recent discoveries suggest these dormant capabilities include enhanced physical regeneration, resistance to various environmental stressors, altered metabolic states, and even expanded cognitive and perceptual abilities, all encoded within your existing DNA. We've identified what appears to be a hibernation pathway similar to that used by bears, Dr. Evans explains. When temporarily activated in human cells, it dramatically reduces metabolic rate and increases resistance to low oxygen conditions, potentially allowing survival in situations that would normally be fatal. Other researchers have discovered regulatory networks that, when appropriately activated, enhance neuroplasticity, the brain's ability to form new connections and rewire itself. These networks appear particularly active during early childhood, when learning occurs most rapidly, but become increasingly restricted as we age. By carefully modulating these networks, we've been able to temporarily reopen critical learning periods, says Dr. Langley. This allows adults to acquire new skills with something closer to the efficiency of children, potentially revolutionizing education, rehabilitation, and cognitive enhancement. Perhaps most remarkably, researchers have identified regulatory systems that appear to influence consciousness itself, networks that modify how we perceive and process our experiences of both the external world and our internal states. These networks don't just affect what we perceive, but how we perceive, Dr. Evans explains. They can fundamentally alter the quality and character of conscious experience, potentially explaining phenomena ranging from flow states and creative insights to meditative experiences, and even aspects of conditions like synesthesia. By learning to modulate these networks, researchers are developing techniques that may allow individuals to access altered states of consciousness previously available only through decades of meditation practice or unpredictable spontaneous experiences. We're not talking about artificial or synthetic states, Dr. Evans emphasizes. These are natural variations in human consciousness encoded within our existing genome, states that have likely been experienced by humans throughout history, but that most people access only rarely or accidentally. The implications of these discoveries extend far beyond medicine or performance enhancement. They challenge our fundamental understanding of human nature and potential, suggesting that what we consider normal human capabilities represent just one configuration among many possibilities encoded in our DNA. We're beginning to understand that human potential is far more variable and expansive than previously recognized, notes Dr. Langley. The difference between ordinary and extraordinary capabilities often isn't in the genes themselves, but in how those genes are regulated and expressed. This perspective shifts our understanding of human development from a primarily external process of acquiring knowledge and skills to something that also includes internal aspects of accessing capabilities already present in our genome. It also raises profound questions about how human culture and technology have shaped which aspects of our genetic potential we typically access and which remain dormant. Modern environments and lifestyles activate certain genetic capabilities while suppressing others, explains Dr. Evans. Our research suggests that some environments might enable access to capabilities that rarely emerge in contemporary settings, potentially explaining historical accounts of extraordinary human functioning that seem implausible by modern standards. This doesn't mean uncritically accepting every historical claim of exceptional human abilities, but it does suggest a more nuanced view that recognizes the interaction between genetic potential, environmental context, and cultural practices in determining the range of capabilities humans actually express. As we develop technologies that allow more direct interaction with our genome's regulatory systems, we gain unprecedented agency in determining which aspects of our genetic potential to access. A profound shift from being passive inheritors of genetic expression patterns to active participants in shaping them. This isn't about becoming superhuman, Dr. Evans emphasizes. It's about becoming more fully human, accessing more of the capabilities already encoded in our DNA, rather than being limited to the subset that our evolutionary history and current environment naturally activate. This perspective frames genetic potential not as something separate from human nature but as an integral part of it, a vast inheritance that we're only beginning to fully explore and utilize. As the sun sets over Cambridge, casting long shadows across Harvard Yard, Dr. Evans pauses to consider the significance of this moment in human history. We stand at a unique threshold, she reflects. For the first time, we're developing the knowledge and tools to consciously engage with our full genetic inheritance, to read the complete story written in our DNA, and potentially access chapters that have long remained closed to us. This journey into the hidden powers of our genome will require not just scientific expertise but wisdom about how to proceed responsibly, balancing innovation with caution, individual potential with collective well-being, and technological capability with ethical discernment. The powers hidden in our DNA aren't separate from us, Dr. Evans concludes. They are us, aspects of our nature that have always been present but that we're only now learning to access and express. The question isn't whether to unlock them, but how to do so in ways that honor our shared humanity and contribute to a future where more of us can flourish more fully. As night falls completely and stars become visible above, Dr. Evans turns toward her laboratory, where tomorrow's work awaits. Another step in humanity's ongoing journey to understand and realize the extraordinary potential encoded within each of us, written in the ancient language of our DNA. Looking back at the history of genetic science, we can see a recurring pattern. Each new discovery reveals that life is more complex, more interconnected, and more wondrous than we previously imagined. From Mendel's simple patterns of inheritance to Watson and Crick's elegant double helix to the vast regulatory networks we're now uncovering, each advance doesn't simplify our understanding but expands it, revealing new layers of sophistication in the genetic code that builds and sustains us. The journey into your DNA's hidden powers is just beginning. As we develop more sophisticated tools for reading, interpreting, and interacting with the genome, we're likely to discover capabilities that seem almost magical by today's standards. Potential for healing, adaptation, perception, and consciousness that transforms our understanding of what it means to be human. Yet perhaps the most profound insight from this exploration isn't about specific capabilities waiting to be unlocked, but about our fundamental nature. We are not finished creations but dynamic possibilities. Beings with vast untapped potential encoded within our very cells, awaiting the right conditions, understanding, and choices to be expressed. Your DNA contains not just the story of who you are, but the many stories of who you could become. A library of possibilities written in the ancient language of life itself, connecting you to both your evolutionary past and your potential future. The truth hidden in your genome is simultaneously humbling and empowering. You are far more than you know, with capabilities beyond what you currently express, connected to all life through a shared genetic heritage that stretches back billions of years, yet remains vibrantly present in every cell of your body. As we continue to decode these hidden powers, we aren't discovering something alien or separate from ourselves, but coming home to a fuller understanding of who we have always been, creatures of extraordinary potential, only now awakening to the vast possibilities written within us.